Good. Okay, so I'm Matt Coelho. I'm a career development fellow in the Cancer Aging and Somatic Mutation program in the Wellcome Sanger Institute. And my work focuses on functional genomics, so using CRISPR-based screens, specifically in looking at cancer variants and what they do, which I'll talk to you about today, um, and also about therapy resistance. So, yeah, as Cass said, questions at the end, unless it's about clarification, in which case, happy to quickly go through that. So what we're actually looking at in the lab is tackling this variant to function problem in cancer. So there's lots of next generation sequencing data of genetic variants that are related to disease, including in cancer. And we're having an overwhelming amount of this genomic data. And actually, the interpretation of those variants is becoming the bottleneck now. So, what those gene variants are doing and if they actually contribute to disease is a really big challenge for diagnosis and for managing therapy. So, in what we call precision medicine, so getting the right treatment to the right patient with the right mutation. And so, if we can actually understand what these gene variants are doing that we detect in sequencing data, not just in cancer, but in other diseases, we can improve these. So, for example, in cancer, resistance is a huge issue, and is mostly driven by genetic changes in the tumor over time, like selection pressure. And so, if we were to actually be able to understand what each of these variants are doing, we would be able to actually get better resolution in terms of patient stratification, so the right drug to the right patient. Also, identify the treatments which might be effective in patients that relapse on therapy. And I'll show you a little bit later about how we can, in cancer, actually design new molecules targeting these drug resistant proteins. So just take a little bit of a step back on this genotype to phenotype question or this variant to function problem. This is one analogy that I like to bring up recently, which is because I cycle to work quite a lot. If anyone's taken a train from London to Cambridge, you'll see that there's this DNA cycle path along the way. And this is actually my route to the Sanger Institute, but it's the BRCA2 gene, which is painted on the pavement. And each one of these is an A, T, C, and G in the different colors. It used to look very crisp around 20 years ago, but now there's lots of mistakes or erosion over time as we age. Similar things happen to our DNA. So if you were to think about what each of these mistakes in this gene were doing, it seems like it would be an overwhelming task because you'd have to look at them individually and test them in a cell assay and see what they do to the gene, A, but also B, what they do to, for example, cancer predisposition in this case. So it's very time consuming and limiting to do this on a one by one, mutation by mutation basis. So instead of doing this at such a small scale, at the Sanger, we obviously do things at a large scale as much as possible. And we've started to use gene editing in these large screens to identify not just one mutation and its function, but thousands of mutations in one assay. And I'll talk mostly today about pooled screening, which you heard about on day one rather than arrayed, which is more a validation, but I should go into a little bit of both. So yes, so we're looking at this problem using functional genomics or CRISPR screens, which is slightly different to the traditional CRISPR knockout screen because that's just disabling the gene function. We're more interested in what specific variants do. So it's actually the ambition of my group to map variants which affect treatment response in cancer at scale. And we use gene editing to do this, multiple different types of gene editing because of the scalability. It's really rapid. It gives you a functional readout, and it's very sensitive. And what I mean by that is you get very rare, the readout of very rare variants and their phenotypes, so you don't have to screen thousands of patients, for example, for rare mutations, which would happen in a handful of patients over time. And also you get mechanistic insight if you couple this to what we call high content readouts, which are different to phenotypic readouts, which is the most basic readout of one of these pooled CRISPR screens. So this is a little bit back to Andrew's talk on day one, the overview. Now go into a little bit more detail. So there's several ways in which you can introduce these variants into the endogenous cell genome using CRISPR. So one is a CRISPR HDR, so homology-directed repair approach, which is what we call saturation genome editing, because we can essentially introduce all mutations at every position using this approach. So we cut the genome and deliver an HDR template for repair, and this encodes your mutation of interest, right? So one issue with this is it's incredibly expensive because you need to make a host of these different repair templates. And it's also difficult to scale because you need so many different constructs for one exon of one gene. And traditionally, therefore, it's been used only for one exon or one gene at a time. It's also is very dependent on high HDR rates. So we need very high levels of homology-directed repair in these cells, and therefore it's mostly been restricted to haploid cells, such as HAP1. But it is incredibly saturating, so you can make all mutations. Base editing is another way of introducing these single nucleotide variants or variants into genes at scale. So you can go here more flexibly across genes because it only depends on the guide RNA. And what happens is this guide RNA directs the Cas9 fuse to a deaminase. Either a C to T or A to G mutation is made at that site. And therefore you can see that just by having guide RNAs, you can have much higher scale. It doesn't require template, it doesn't require HDR. And also you can look at multiple genes at once. So it's a little bit cheaper, and also it doesn't require haploid cells. So we actually use this quite a lot because we work on cancer cells which need to be responsive to certain drugs. So they may actually have multiple copies of the oncogene and actually sometimes aneuploidy as well. So that's pretty efficient, but less saturating than the SG approach because there's only certain numbers of mutations you can make and it requires this PAM sequence. Lastly, most recently is the prime editing machinery, which is much more flexible. So it can introduce essentially any mutation, which is less than 50 nucleotides in length. Again, super flexible because it doesn't require HDR, it doesn't require template. And the one issue with this is that it is quite inefficient. And at the moment, the pegRNA design rules or prime editing guide RNA design rules are like quite poorly understood. But that's improving, and we can talk about that offline if you'd like to use this with higher efficiency in your labs, for example. One thing to note here is that this is a reverse transcriptase, which actually directly transcribes the edit into the cell. And that's how that machinery works, which again is quite remarkable, as Andrew mentioned in his talk. Okay, so they all have their advantages and disadvantages. I'll talk mostly about base and prime editing today, but I'm happy to talk also about SG approaches if it fits your interest. We're more interested in drug resistance. So you can use prime editing with higher efficiency, again, if you have haploid loci. So that means one copy, similar to what I mentioned with the saturated genome editing. And that's one way of boosting the efficiency, especially if you need to have homozygous edits for your effect. And that's something that we've looked into. For example, this saturation prime editing paper actually used prime editing on this NPC1 locus, which is related to a neurological disease, not cancer at all. And this was actually the first use of prime editing as a saturating screen to look at lots of variants on one gene. And this is the reference here, but actually what they did is a clever little strategy to get rid of there's 3 loci in these cells, get rid of the other 2 loci to just leave 1, so they could get just 1 copy edited with prime editing, which is much easier than 3. And the way they did that is to use a system that we developed, which is to very specifically cut 2 alleles which are wild type for these SNPs. So this is a technique called CRISPR guard, where we can actually get Cas9 to cut very specifically and discriminate with a single nucleotide variant here, a SNP in this allele. So it cut these 2 alleles, but not this one because of the SNP here, and therefore you get this haplodization, which you can deploy to any of your genes of interest if you can identify that there's a SNP on one allele and not the other. So very briefly, how this technology works to increase Cas9 specificity is it uses short guide RNAs, which don't cut, because you need about 20 nucleotide guides in order to cause double strand breaks. So what happens with the short guides is that they just sit on the DNA and have a nuclease dead Cas9 there, essentially. And so they protect the sites from the actual guide, and they protect the sites from the nuclease active Cas9. And so you can program these to your wild type sequences here. The short guides will bind, protect these sites, and then the long guide will come in, protect here, and then the long guide will come in and cut these wild type sites and leave you with a haploid loci. So that's one way of getting around the low efficiency of prime. There are other strategies which we can go into later. I'll talk to you about those in this talk. So base editing, just very briefly, because I know it's been introduced already, it's a little bit different to Cas9. It depends on the Cas9 protein, but it actually has a fusion of either a cytidine deaminase shown here or an adenine deaminase. And so this directly deaminates the cytidine to make uracil, which is by replication and repair incorporated as a thymidine, and so you get a CTT mutation. And this, all of these constructs are available from Adgene, and indeed David Liu's group in Harvard has developed essentially all of these reagents I'll talk about today. So prime editors, cytidine-based editors, and adenine-based editors. So the adenine-based editors, again, are useful if you want to make an A to G mutation instead of the C to T. And what's really exciting is that these are being used for medicines. So they're being used for gene editing medicines for monogenic diseases, such mostly blood disorders, but other neurodegenerative diseases and things like that are being trialed at the moment. Indeed, patients in the UK are already being treated with base editors, for example, in CAR T therapies. And the reason why is because they're safer, because there's no double strand breaks. So double strand breaks can be quite toxic and lead to lots of rearrangements. These only cause a nick, and therefore they're seen as a safer alternative, also quite precise. So we've been using these not just to make one mutation like you would with a therapy, but making thousands of mutations to look at what they do across the genes essentially, and how they affect cancer cells. So I actually encourage you to read this review on functional genomic screens, which we wrote a couple of years ago, which actually outlines all of the steps of designing, carrying out, and analyzing, including quality control of a functional genomic screen. And it also talks about other applications like coupling it to single cell RNA sequencing. So this is actually quite an in-depth review, which lots of people were involved in writing, and we can send that around in the chat later for further reading. The base editing actually can be a bit heterogeneous. So sometimes the cells do base editing, and sometimes you get a cell line where the base editing is not as efficient, or for some reason your base editing machinery might not be as efficient. So one way to actually track to see if your base editing experiment is working without doing very expensive sequencing, Sanger sequencing or next generation sequencing, is to use a reporter. So we actually use reporters a lot, fluorescent reporters, in order to just sanity check the experiment and make sure that the editing is being carried out in the cells and your machinery is working. So these are really useful things to have in the lab. So this one is a transgene, which is a blue fluorescent protein, and actually one nucleotide change from a CAC histidine to a tyrosine causes this BFP to GFP transition. Remarkable that it's just one nucleotide that can do that, but that fluorescent shift can be monitored with microscopy, can be monitored with flow, and here I'm showing a flow plot to show that the BFP has been made into GFP. I just want to warn you actually that the fire alarm might go off in a moment as a drill. So if I stop for a moment, that's probably why. And so that's actually useful to sort those cells which have been edited and also to test new base editor constructs and also track them with microscopy, for example. So we've been using that a lot. And again, the constructs have been published and you're welcome to use them in your lab. The sequences are out there. We do similar things with prime editing, and I'll tell you about that in a moment. The prime editing machinery, just to go into a little bit more detail, is again a Cas9 nickase. So it creates a nick so that the unedited strand is essentially repaired using the edited strand as a template. And this is an approach which has been used by David Lee's lab in the base editing as well. The reverse transcriptase essentially, once the guide is bound, it will copy the pegRNA reverse transcriptase template, and it will copy into this flap which is formed, the cDNA essentially, which is made from the reverse transcriptase. Then after that happens, the reaction is in this state here and point into the right direction, into this intermediate state where it could go back to wild type if the edited flap is excluded, or it can go back to the edited state if the edited flap is included here. And that's actually catalyzed by this DNA repair machinery. And the details are in this Nature paper here. These constructs are also available from Adgene. I should say now that the constructs have come a long way and there's been several iterations of improvement for the prime editing machinery. This is actually prime editor version 2, which I'm describing in this paper. I would recommend using prime editor max, which is been optimized for expression, like codon optimization, and also using knockout of mismatch repair. So for example, MLH1 knockout to make sure that the edit is kept in the cells and not repaired back to wild type. So those are the two things to the editing machinery. And finally now, David Lee and Britt Adamson's lab has included a hairpin at the end of these pegRNAs to protect them from degradation. Because actually what they found is that because these are really long single-stranded RNAs, they're actually degraded by exonucleases normally in the cell, and that actually reduces the efficiency quite a lot. So we normally add a little hairpin called an Evo Pre-Q at the end of these to protect them. Okay, I hope that's all clear. So we're now using these machines to introduce mutations in cells, and I'll show you a little bit about the screens we've done. So this is like a traditional CRISPR knockout screen, but instead of just looking at gene level effects, we now look at variant level effects. So what we do is we introduce a lentiviral library into cancer cells in this case. And generally we have a selection pressure. So that can just be the growth of the cells. In this case, we added interferon gamma, which is a cytokine. And these cancer cells are sensitive to it. So it causes cell death. And then we did knockout screens, and then we tiled the interferon gamma pathway shown on the right with cytosine and adenine base editor. And this is actually really important in cancer because we're interested in cancer immunotherapy. And T cells normally pump out the interferon gamma, and this causes cancer cell death. So if you were to lose function in these genes, you would actually be resistant to interferon gamma and also resistant to T cells, probably resistant to these therapies called immune checkpoint blockades. So we screened across these genes, which we identified in the CRISPR-Cas9 screen, essentially by guide RNA sequencing. So what we do is we introduce a library and then we look to see which guides go up and which guides go down at the end of the experiment versus the control and versus the library. And then we can look for resistant variants encoded by the guide and also sensitizing variants encoded by the guide. And that's what I've shown schematically here. And then we did some arrayed screening, which I'll talk to you a little bit about, in organoids with T-cell co-cultures, which is a little bit difficult to scale. So we generally go from high scale to lower scale when the complexity of the screen goes up. So this is what these types of data look like, these base editor screens. You might have seen something like this before. Essentially, on the x-axis, I'm showing now the position, the amino acid position of the JAK1 kinase, which is involved in from gamma signaling. And on the y-axis, I'm showing the z-score, which essentially is like a log fold change, which is normalized for the standard deviation of the data. And this is the log fold change essentially of the control guide RNA read counts versus the interferon gamma treated condition. So if anything is above the 0 mark, we get enrichment, and therefore it's a loss of function and resistant. Anything below 0 is gain of function, and it's causing more death and more JAK1 signaling. They're obviously rarer. And so what you can see is actually some of these missense variants are incredibly deleterious, as deleterious as stop codons, which are shown in the diamonds here, and splice variants, which are shown in the triangles. So these are really strongly inhibiting JAK1 function. And you can see actually, if you map these back to cancer genome sequencing, going back to the first slide, these variants actually have been found in patients which have been resistant to immunotherapy. So this is actually now linking the variant that has been found, a variant of unknown significance clinically, to functional data to show that it actually is causing a loss of function in this protein. And that might actually be causing why these patients have been resistant to this therapies, causing the immune system to attack the cancer. So this is really important structure function data as well, because the JAK1 crystal structure, the full-length crystal structure, hasn't been experimentally solved. Obviously, there's predictions, but we can get these really nice insights into protein-protein interactions, which are hard to predict from these genetic data alone. So, for example, the kinase domain here at the C terminus, we found this patch of gain of function mutations, and what we actually thought from mapping the 3D data is that this is mapping to a protein-protein interaction, and that's what you can see here. Okay, so this is in red, these residues I showed you, and they actually map to a protein-protein interaction with the JAK1 kinase with a SOX1, which is an E3 victim ligase. So we can use these data to actually identify protein-protein interactions, which normally cause JAK1 to be degraded. And here in blue are these really strongly loss of function mutations, which we map directly to the ATP binding site and active site. So this is really useful for potentially drug discovery, as well as understanding resistance to therapy. So that was all on proliferation screens. So we're just measuring guide RNA abundance by next generation sequencing. We don't really get an insight into what each of these variants are doing to the cell, but we can actually do that with single cell transcriptomics. So that's what we call perturb-seq. So for a really more mechanistic insight or an in-depth understanding of what each of these variants are doing, we don't just want to understand if they're causing increasing growth, we want to understand why. So we can look at the signaling going on, and the way we can do that is by 10x transcriptomics, for example. So what we've done here is a follow-up study on that cytokine resistance screen. And instead of just reading the guide, we've now mapped that also to, in the same cell, the transcriptome. And the way we can do this is to read out the guide RNA in the transcriptomics. So in the same cell, we get the transcriptome and the guide, which is also barcoded. And so you get these nice UMaps, which is essentially dimensionality reduction of these transcriptomics data, and hope you can appreciate that lots of the loss of function mutations we've identified in JAK1 cluster very distinctly from the wild type cells or the non-targeting guides. This is purely because they have completely different transcriptional response to interferon gamma now. So these ones don't respond, these ones do. And actually, if you can do DNA sequencing on the same single cells using the Mission Bio machine, you can actually identify exactly what the mutations are in each of those cells and verify that they're there. So not just using GIDA as a surrogate for a genotype, actually verifying what the genotype is through amplicon sequencing. So here we can show that the stop codons we've introduced lie in this cluster of cells which don't respond, splice variants, and then missense variants, as you might expect, which are these protein amino acid to amino acid substitutions. They're either benign, which is here, or causing loss of function. So we can actually identify mutations which are introduced that don't do anything for the signaling, which is really useful for example, clinical diagnosis and things like that. So it's not just so binary with the single cell transcriptomics, but the beauty of this single cell transcriptomics from CRISPR screening is you can get more nuance in the actual mechanism of action of each of these variants. And so what we actually identified is some cellular heterogeneity. So what I mean by that is some of the cells with the same variant shown here in the box responded to the interferon gamma and some of them didn't. And so this is purely because these variants are kind of semi-penetrant, or what we called separation of function variants. So with the same genotype variant, there's a bit of a profile of heterogeneity in each of these cells. So sometimes they're responding, sometimes they're not. And here are the wild type and the complete loss of function cells on either extreme. So with the proliferation screens or a FACS screen, for example, we would never have identified these variants as having a different mechanism. Good. So that was a little bit more of a complex readout, high content readout on the CRISPR screen. And I think over time, more of our experiments will transition to these, because at the moment they can be quite expensive, but they definitely offer a lot more insight into how these variants are working. And the same can be done for CRISPR knockout screens, for example. Just to move on to therapy resistance to targeted drugs in cancer. So actually we wanted to, instead of looking at interferon gamma signaling, we want to also look at resistance to molecularly targeted drugs in cancer. And the aim is to actually eventually predict the resistance to therapy, because as I said, these are often genetic and often there's no backup therapy. So it'd be really useful to actually get a handle on which patients are relapsing and why, and how we can treat them better. So in this study, which was published last year, we looked at the 10 molecularly targeted drugs in cancer and the resistance across 11 cancer genes using cytidine and adenine base editor screens. And so again, we were screening using lentiviral libraries of guides, tiling these genes. And actually, Jansu will tell you a little bit in the practical after this session about how you design these guides for libraries to do your base editing screens, for example, using this tool called Bestimate. So we then treated the cells either with nothing or with vehicle or with these 10 drugs, and then we compared the resistance to each of these in turn. And we also did perturb-seq, so single cell transcriptomics, to look at the mechanism. What we were really interested in resistance variants, but also sensitizing variants as well to the drug, because the sensitizing variants could be useful to identify patients which would really respond well to therapy. So this is a little bit of a busy slide, but I want to show you the power of these screens and the kind of scale of these screens to kind of hopefully get you excited about using base editing in your applications as well, not just for cancer, but for anything you're interested in. So in this graph, each dot is a different variant, which we introduced with base editing. And the color represents each gene. So here you can see the 11 genes we've looked at. So again, the power is we look at the endogenous locus, we look at a cancer cell setting, and also we look at not just one gene, but here multiple different drug targets. And we can look at the variant effect in each case. So the y-axis is the presence essentially of this MEK inhibitor and the growth of the cells in the drug, and the x-axis is the growth of the cells essentially in the absence of drug. So anything off the diagonal has a different growth rate in the presence versus the absence of drug. And so what we can see is that these variants cluster into different categories which affect resistance and response. So here is this what we call canonical resistance. So this is actually having a resistance effect only in the presence of drug. And so that's actually on the MEK1 drug target. So if we were to look at these, actually they're found in patients, these variants, and they map directly to the drug binding pocket. So that's quite an obvious way of getting around inhibitor binding. The second class that actually have we call drivers because they have a growth advantage in the presence and absence of drug. And these are actually known driver mutations in the PI3 kinase gene and AKT. These are really interesting sensitizing mutations because they actually cause the MEK inhibitor to work better. And they're actually loss of function mutations in EGFR. And when you combine EGFR inhibition with MEK inhibition, you actually get a stronger effect. So this is a good nomination of a combination therapy. And finally, these drug addiction mutations. So these are quite strange because they, in the absence of drug, cause the cells to grow slower. And in the presence of drug, they cause resistance. So what's happening is they're really activating the MAP kinase pathway and they cause the cells to stop growing because in the absence of drug, the activation of the pathway is too strong. So there's a threshold for the right amount of oncogenic signaling, which is quite fascinating. So you can imagine if you take the drug away, the cells stop growing, and in that drug addiction mutations, that seems to be the case. So in black here, we took away the drug, and the cells slowed down in their growth. So for those patients with those variants, it probably would be better to give them a pulsatile therapy, so they kind of have these holidays in treatment. Those resistance mutations would go down. So we can link that to the single cell transcriptomics again, and indeed that matches up with the proliferation screens. So we can see here that each one of those 4 classes of mutations are represented essentially apart from the sensitizing mutations, which we didn't include here. But what we can see from this clustering of all the transcripts from those cells with the different types of resistance mutations is that with the drug addiction mutations in red, they actually have a much higher level of activation transcriptionally of these MAP kinase pathways. And that's shown here in this histogram as well. So in the presence of the MEK inhibitor, they have a, sorry, here is other treatment, but they have a much higher level of MAP kinase signaling. So the proliferation screens and the transcriptomics marry up quite nicely, showing there's complementarity there for both approaches. But we also find some unexpected pathways which are turned off in those drug resistance cells as well, like the interferon gamma pathway. So I just want to quickly talk to you a little bit about how we can use these data to inform clinical decision making for patients which have relapsed on treatment. So one treatment for EGFR mutant cancers is this drug, gefitinib. And actually the most common mutation in 50% of patients treated with this drug is this T790M mutation, which is in the EGFR protein itself, which is the drug target. And we found this using the base editing screen, as you can see here, using the cytidine base editor. And so we can immediately and very rapidly find drug-resistant mutations which are clinically relevant. And actually, this is important because these second-generation inhibitors, for example, osimertinib, developed by AstraZeneca, target these resistant proteins. So if you're a patient and you find you've stopped responding to gefitinib, you have this mutation, you'll be given a second drug which should be effective, called osimertinib. And so finding these mutations early helps us to kind of prioritize these proteins for drug discovery, but also accelerate the development of these second line inhibitors. Needless to say, resistance to osimertinib also develops, and we actually found some mutations in EGFR and PI3 kinase, which cause resistance here. But we also found drug sensitizing mutations as well. So again, prioritizing patients for the right therapy in sequence is important, and we found the same thing in part one, which is a target of olaparib and niraparib, mostly for breast and ovarian cancer. And we were finding these drug sensitizing mutations, but also drug resistance mutations for one inhibitor, but not another. So again, if you're resistant to niraparib with this variant, you might be responsive to a second inhibitor, which is already out there, olaparib. So this type of structure function information is really useful for drug companies to, we think, prioritize patients which would respond better, such as those harboring these cancer variants, and also respond to another inhibitor which is already out there if you have a resistance mutation, for example, to niraparib here. So we amalgamated all of these data together, and we kind of built a variant function map to outline all of the resistance mutations which are present in each of these drug targets using our base editing screens. And we found several interesting things throughout this pathway. For example, we found mutations here in PI3 kinase, which actually give resistance to multiple drugs, but could be treated with an alternative inhibitor, which is already out there and approved. So a second line therapy. Again, in part one, we showed that if you're resistant to niraparib, you could potentially be still sensitive to another agent, olaparib. We also found these biomarkers of response. So especially in EGFR, we found these drug sensitizing mutations from the base editing screens, which identify patients perhaps which would be really super responders to EGFR inhibitors and therefore should be enrolled in these clinical trials, for example. And with the drug addiction mutations, we found variants where they overactivate the pathway. And so if you take them off drug, those cancer cells should stop growing. And those are called drug holidays, for example. So alternative scheduling of inhibitors. So we all hope that this could inform some clinical management of drug-resistant disease in cancer. So the base editing screens can't discover all variants, as I mentioned, because of the saturation. And so one thing that we're moving towards is using the prime editing screens in conjunction with the base editing screens in order to get higher resolution maps of variants and also to introduce mutations that we cannot introduce with base editing. So as I mentioned, prime editing is a little bit harder to get working because of the efficiency issues and because of the prime editing design of the guide. But we are actually getting really nice data from these prime editing screens now. And so just to say that the C797S mutation is found in patients which relapse on this EGFR inhibitor osimertinib, which is those second line inhibitors which I mentioned. We can't introduce this mutation with base editors, either the C to T or the A to G base editors, because it's a transition mutation. Sorry, it's a transversion mutation, not a transition mutation. So the transition mutations can only be introduced with base editors. And so to get that mutation, we have to go to prime. And that's what we've introduced here. So we followed up on the base editing screen with prime, and we could introduce this mutation. And sure enough, it's incredibly resistant to osimertinib. And we also find other depleting variants as well. So we actually went after another reporter and developed a fluorescent reporter, which is very similar to the BE flare reporter, and we've called this PE flare for obvious reasons, to track cells which do prime editing and actually improve prime editing efficiencies. And that's shown here. So in this case, this is unpublished actually, but I'm happy to share the sequence with you if you're interested in in using this reporter in your labs. But we have an M cherry with a stop code on, and therefore only when you introduce this T to G mutation, again, impossible with base editing, can you actually reinstall the glutamic acid and you get read through and you get M cherry positive cells. So we've now been using this to sort for M cherry positive cells and test the prime editing efficiency in our cell lines, and it works really nicely. I'll show you here recent work from Francesca in my lab. We've used these cancer cells with MLH1 knockout, which is this mismatch repair machinery, to get the prime editing levels higher. As I mentioned, that's a trick to improve efficiency. And you can see here, we're showing GFP is a marker of the prime editing machinery. This is the control in this flow plot. And this is M cherry marking the prime editing of this PE flare construct. So without docs, we can see some leakiness, but we get some low level of editing. And then when you induce more expression of prime editor with doxycycline, you get about 25% editing. And we've now been able to single cell clone really high editing cancer cells to up to about 50% prime editing, which is no mean feat actually, because people are reporting lower efficiencies by and large. So we think this is a good co-enrichment strategy to help you with your prime editing screens as well. So just to finish up on how we can use base and prime editing to inform drug design as well. And if you're interested in structure function information and not just genetics, this might be more interesting for you. But we've been very interested in using structure function information with drug companies, for example, GSK in this case. And in collaboration with Matthew Garnett, we've been able to identify in proteins, in this case, the Werner helicase, very essential domains in these cancer genes, which if you mutate them, cause the cancer cells to die, and therefore could be good drug targets. So we already know that this protein and gene is a good drug target, but we don't know exactly where to inhibit it. And using these mutagenesis screens, here is base editing screen, we can identify here that the helicase domain is super important, and therefore actually the drugs which are all out there now in patients from GSK and other companies all bind to the helicase domain and inhibit helicase activity. Because at the beginning, it wasn't really sure which one of these functional domains was actually relevant. So we're interested in doing this type of work to look at drug discovery and also hone in on which parts of the protein are actually tractable for inhibition in cancer therapies. So just to summarize now, essentially what the use of these gene editing screens, and we call them multiplexed assays of variant effect, or MAVES, just to show you that the use of these multiplexed assays of variant effect in cancer diagnosis and treatment. Hopefully I've shown you that we can use base editing and prime editing to identify very rapidly drug resistance variants in cancer to immune checkpoint blockade, so these immunotherapies, but also targeted drugs. And so that could have a step change in how we look at resistance, because at the moment it's very retrospective. We look at cancer biopsies and they can be very limiting. So we can really look at especially investigational drugs early on and the resistance mechanisms using these screens to get ahead of the curve even before they emerge in patients. Secondly, I didn't show you data on NTRK1, but we can, in the Nature Genetics paper, I hope I showed you that we can nominate effective second-line therapies. So those variants which cause resistance to one agent, still sensitive to another, and these screens show that, so we can actually hopefully offer a patient with resistance another option for treatment which is already licensed and out there clinically. And thirdly, we're using this more and more, these screens to link genotype to phenotype and structure to function, and therefore using it to guide drug discovery, especially for drug-resistant proteins, and for where we don't have a full handle on which part of the protein is driving the oncogenic function, for example, a functional domain. And in that case, the Werner helicase was the example where drug discovery is ongoing and clinical trials have started. So that was a whistle-stop tour of our work that I'm involved in using base and prime editing. Looking forward to questions. I'd like to also thank my team. It's a big collaborative effort, shown work from everyone here. I'd like to thank funders, Cancer Research UK, and we have a job opening actually in my team and BaseRx, which is a company spin out from the Sanger as well. So if you're interested in machine learning and computational science and you found this work interesting, then please reach out to me or your networks and spread the word. And yes, and I'll stop sharing now, and hopefully there'll be lots of questions and discussion. Thank you.
I have a question or maybe more of a clarification. First of all, thank you so much. That was really interesting. So you were saying about how you can use kind of finding these variants to map protein-protein interactions. I'm just kind of wondering how you go from looking at the variants to then looking at the protein-protein interactions, and then kind of, I think you mentioned um, like the E3 ligase is causing degradation. I'm just kind of wondering like what kind of follow-ups that you would do if it's all computational or if you go on to do more kind of, yeah, experiments.
Hmm. Yeah, thanks, Bronnie. Good question. So actually, one thing we can get purely from the genetics is and the design and annotation of the guide library. Actually, Jansu will be teaching you a bit later. We'll go into it in a bit more details, but we use lots of pre-existing databases to map where we think these variants are on the protein in 3D. And there we have several annotations such as predicted protein-protein interactions, the domain that they're in, and so we kind of superimpose all of these annotations onto the screening data already. So there's some predictions. At the moment, it's a bit harder to map everything onto the 3D structure, and there's no, we haven't implemented a way to do that in detail yet ourselves. There are certain tools which are out there. One is genotype to phenotype from the Broad Institute. Again, I can send that around later, but there's a tool already out there where you can map it onto the structure. Also, there's alpha missense and AlphaFold, which we use to kind of look to see where the variants are and how the protein change may, as a prediction, affect the folding or interactions intramolecularly. A bit harder to do with protein-protein interactions. As for validation, I think it's really important to validate these screens, especially from base editing, because we're not 100% sure what the edit we've installed into the genome is. It's only a prediction unless we do amplicon sequencing. And so one thing that we're doing is to sequence our hits afterwards to check what they are. And then we do smaller scale validation screens, which may include biophysics or classical biochemistry and things like that, like protein-protein interactions. We generally don't do lots and lots of small scale experiments because that's not really what we're geared up to do at the Sanger, but we hopefully, have hypothesis generating research and people can look into that in a bit more detail. Yeah, so we definitely follow up with the validation. I can talk more about the genotype validation a little bit, but we're using also these sensor assays where the guide edits a synthetic sequence in the plasmid, so we can actually have a surrogate measure of the editing for each guide.
Thank you so much.
In primating, the primer binding site is complementary to the guide RNA, which can cause self-hybridization before donor recognition. We often need to test several guide RNAs to find an individual one. The primer binding site should be complementary to the not the guide itself, but it can be complementary to the guide. Normally complementary to the target sequence-ish. We often need to test several guides to find an efficient one. Do you face the same, or do you most of your designs work well? Which base editing? Okay, so just to address that first, certainly I would recommend designing multiple peg RNAs per variant because we're still not at the stage where we have like a Cas9, a very high confidence guide. So we generally are doing something like 5 guides per variant to make sure we get one which works well, to give more confidence. Some people in literature have gone up to 20 peg RNAs per variant, but obviously that can be prohibitively, well, with the scale, it can be prohibitive depending on how many variants you're looking at. We don't empirically design our guides. We use Predict, which is a machine learning based output of the Mathis et al. Paper from Nature Biotech, where they basically looked at editing rates across thousands of peg RNAs and built a machine learning algorithm on the data to generate the best prime editing guides using their sensor editing system, which I just mentioned. And therefore, we have pretty good designs computationally. And so we just take those, and some of them work, some of them don't, but we don't have a good idea of percentages yet. But generally, from the paper, again, I can send it round afterwards. I'm making some notes. Generally, they're reporting much better efficiencies than if you were to do it empirically. So which base editor version are you using, and which gives you the best efficiency with minimal bystander edits? So we generally go for to increase saturation, the NGN PAMs. So that's the Nurecki lab Cas9 variant, which instead of binding like SP Cas9 wild type NGG, it binds to a flexible NGN. And so that gives you obviously much more coverage throughout the genome. And the base editor variants are generally base editor we call 3.9, which is from the cytosine base editor from John Dench's team, and the adenine base editor is ABE8E, which is from David Liu's team, which is highly efficient. The bystanders we don't really care about that much. We're more interested in better efficiency. If you were to really worry about precision, it's more about the therapeutic genome editing. You could use some of the ones that David Liu's lab has engineered, which we tested but have much lower efficiency. So it's kind of a trade-off between precision and efficiency, and we generally favor efficiency in this screen. Hope that answers your question. And is SGE data publicly available? And if so, how do you access it? Fantastic question. Another link to send around is the MAVE database. Okay, so MAVE-DB is a repository of all of these screening data, and from SGE, base editing, prime editing, mostly SGE, and also deep mutational scanning, where people overexpress cDNAs, libraries of variants, which is probably by far the most common data set for these types of variant function screens. So there's a huge catalog of those. And if you're interested in certain proteins, I recommend looking at MAVE-DB. Thank you, Bo. Okay. Please do send paper. My institute doesn't have access to the journal, sadly. Okay, so I can send PDFs. Thank you. What about KRAS G12C and G13D mutations? Any findings related to this variant in your study for drug sensitivity? From Zainab. Yeah, thank you, Zainab. That's great question. So the models that we actually use for the study, the H23 model is actually G12C already. So it already harbors that mutation. So that's why it's sensitive to sotatercept and adagrasib, which we used in the study. Um, yeah, so we found other mutations in RAS which gave resistance, and other MAP kinase proteins, for example. So they're pre-existing. So one thing you should always bear in mind is when you start mutating your cell line of interest, what is the underlying genotype? Because if you design your library, most of the time you take the ensemble sequence, which is the consensus wild type. So one great thing about Janssi's tool, which you'll learn about, is that you can not just deliver a wild type reference sequence, but you can design your guide against a mutated sequence, which might be, for example, a genetic disease you're trying to correct, like sickle cell, doing base editing, or for example, you're trying to mutate your mutated oncogene and you're interested in correcting G12C, for example, Zainab. So hopefully that answers your question there as well. But very good point. Any other questions? People can feel free to raise your hand as well. They're not in the chat. Okay, let's go with Mehmet.
Morning, Matthew. Thank you for the lovely presentation. You mentioned that going through different genome editing to your cell lines to create to find out it causing cancer types, so modeling cancer models. And also you mentioned that you detect proliferation to see the level of proliferation going down because that's the ideal way. So what's the standardized proliferation marker you used throughout your essay? And how did you, is it like you detected like weekly base or daily base? And what is the specific method? So if you
Yeah,
enlighten me on this matter, I would fully appreciate.
absolutely. Yeah, thanks, Mehmet. So the pooled screens, we actually deliver all of the variants in lentiviral plasmids, integrate them into the cells in a pooled way, and then we measure the abundance of each of those variants by next generation sequencing. Essentially, we PCR at the end, genomic DNA extract, and then PCR the guide. And so when you look at proliferation screen after several weeks, you look for the guides which have increased in abundance versus the time zero, which is the ones which increase proliferation, so through clonal selection. And then the ones which decrease or deplete to zero after a couple of weeks versus the time zero, they're the ones which cause, cell death or stop the cells from growing. And so with this depletion after cell doublings is what we look at is purely by sequencing. So we're not doing any assay for cell proliferation. It's purely counting. And if we were to do follow-up experiments with a RAID, we would then introduce the guide. We're interested in a single well, and we might do something like a traditional cell titer glow assay or IncuCyte curve or something like that. So hopefully that clears it up for you.
Yes, it's quite clear because I was just curious to know if you use any like immunofluorescence based, I mean fluorescence type marker like Ki-67 or pCNA, proliferation cell nuclear antigen, that kind of marker using fluorescence microscopy or using flow cytometry type of
Yeah.
application. That's why.
Yeah, so that we could sort by staining for Ki-67 and things like that, but essentially all of these readouts are purely by sequencing. So if it's a selection marker like, Ki-67, you could do it, but by fax and then sequencing. But yeah, you can't deconvolute the guide variant purely by microscopy in such an easy way. That requires, yeah, because you could see which cells are proliferating, but you don't know their genotype until you sequence them. Okay, good. Thank you.
Thank you.
You're welcome. Just to quickly answer one online. Are the barcoded guides commercially available? No, they're not commercially available. I would recommend barcoding your libraries using PCR method with a degenerate primer. So from Sigma, because the Twist, for example, and IDT, they're not very good at designing, or they don't allow you to design libraries with NNN complexity. So you can just get from Sigma, for example, an NNN degenerate primer and then amplify your library, and they'll introduce a barcode in that way in a very cheap fashion. And you have a very complex library which is suitable for example, linear tracing, or for example, perturb-seq. There's a question from Marina.
Yes, thank you, Matthew. That was a very interesting presentation. I had a question. So I was interested by the application, or where you see the application for the base editing or prime editing, because you said it can guide also the novel drug design, right? As you showed with the Werner protein. How do you see it for, let's say, challenging drug targets that where, for example, you don't have a crystal structure or a transcription factor, for example? Have you like tried it also on, let's say, targets like this that are challenging? And do you see that it could definitely be like an advantage of using such a screen for such a target in that sense?
Yeah, I think it's a really good question. I think that's exactly the kind of application that we hope to make best gains, because where there is a lot of structural knowledge already, obviously, we kind of know the protein-protein interactions and intramolecular interactions, but actually, overwhelmingly, we kind of have domains which are crystallized in a certain conformation in isolation. And it's just a static picture of what's going on. So, for example, with the JAK1, it was a bit surprising that most of the mutations we found were in the pseudokinase domain to actually stop the function of the protein. And that's not the kinase, right? So there's a very complex picture of inhibition, intramolecular inhibition. And you certainly will find unexpected things here, which we probably wouldn't find with just looking at the kinase domain alone. And also, we're still behind with the AI on mapping protein-protein interactions. I think the multimer is still not super ready for prime time yet, and you can't access it for drug discovery in most cases anyway, because it's behind certain restrictions, let's say. So yes, I think the structure function information from genetic screens is very powerful. Hopefully it'll be useful in drug discovery. Thank you.
Yeah, thank you.
The Marcos says what's the representation of the scale of the library in these experiments? Yeah, so that's thank you for raising that point. The scale is still the limiting factor. So it's Alice Refermat talked about the number of cells you require for looking at a certain number of guides, and how you need to have multiple cells in your screen with the same variant or the same guide RNA perturbation in order to get a reproducible phenotype. So that might be, for example, traditionally around 1,000 cells, or what we call 1,000x representation per guide. So if you think about scaling that up to, 1,000 perturbations, we have 1,000,000 cells, and it scales. So the tissue culture experiments become huge, and that's one limit for the cost and also the practicalities of actually doing the screen. The base editing screens we consider to be quite large. That was 32,000 variants that we looked at in a programmed way. So that's already pushing the boundaries of what one person can do in tissue culture hood. We generally use these multi-layer flasks. So we culture. I can show you a picture of some of our incubators, but sometimes we have multiple 5-layer flasks, and each one has around 250 mils of media in it. So again, the costs go quite high. So it's all about how much of a sadist you are and how much money you can chuck at it. But we're really waiting for a little bit of a technological advance in order to go from a couple of genes to hundreds of genes and whole genome. And, I think actually you might learn a bit more about that. Some people who are going to the next course on Cas12a and things like that, where you can have multiple guides in the same construct.
Thank you, Matthew.
Hello, thank you so much for such a great insight, and I'm really looking forward to this paper, and I will surely read this paper. My question is, since I'm also working on CRISPR screen, and obviously it's a new introduction to me as well, and I'm performing here on colorectal cancer cell lines. My question is, I've seen studies comparing time 0 versus time 14, like they culture cells for like 2 weeks. I was thinking, what if we can do early time points for genomic DNA extraction compared at early time points, let's say just a week, or it needs to be time 14? Because I'm reading papers and they have been doing for like 2 weeks. What do you think if we extract early genomic DNA for the CRISPR screen, or it will have an impact or no? I'm just curious.
Yeah, so there's no right answer, Zainab, to this because it depends on your phenotype. So some perturbations, let's say, because it could be mutation or knockout or whatever, they'll have a very rapid effect and there'll be very strong effect, and that will be happening after even a couple of days. Some of them will have much weaker effects and therefore and also a slower effect, especially to do with the gene turnover, right, as well, of the protein. So for those effects, you will need to culture the cells for longer. And so what I recommend doing, if it's possible, is to take as many time points as you can feasibly do throughout the screen, because these screens are a lot of work. So what you don't want to do is to do the whole screen and then have to repeat it because you've taken the wrong time point, or you need to longer. So what we generally do is take lots of conditions, and then in the analysis we can say, right, some of those genes weren't fully depleted at day 7. Day 14 looks much stronger and cleaner. We use day 14. Some of the SGE papers have actually looked at multiple time points and found that some variants deplete quickly and some deplete slowly. And so, yeah, it's very insightful. Yeah. You get more data back for the same amount of work.
Yeah, thank you so much. Thank you.
You're welcome.
As a follow-up on Zainab's question, for the screens, actually, I myself did this last screen where we have done time course. We have like 10 days and 14 days. And my question is more for the analysis part, is that the pipelines of analysis now, where the bioinformatician is actually working on now, is that it's either, and we had actually a drug challenge in addition. So we have 2 lines on the screens and with time courses. So it's either the analysis that we could find that's for the bioinformatic part, is that either for different time course with only 1 line, or is it with or without the drug with only 1 endpoint? So maybe we will see that later on in the course, but I'm just curious to know if there is actually actual pipelines for the analysis of this type of data in this with this experiment setup.
Yeah, so yeah, thank you, Naima. There is actually, it should be super flexible. You should be able to do all of these, and I recommend you play with the data as much as possible and then do lots of comparisons. On Thursday, tomorrow, there'll be an analysis session run by Samin and Jamie to look at pooled screening data with MAGEC. So this is the kind of statistical framework, and essentially you can make every comparison. Mostly our bioinformaticians give you every comparison, and even if you haven't asked for it, then you have to you have to look through.
That sounds great.
And it's so it's once you've got the data, I recommend doing all the comparisons, because you might end up finding one which is surprisingly informative. And actually you can do lots of things like cluster them using principal component analyses and check for correlation between different conditions and things. So yeah, that shouldn't be the limiting factor.
I felt like I made it too complicated for the bioinformatician at the end of the day. As a biologist, I wanted to cover up all the conditions kind of to have it as enriched as possible, but then it was a bit challenging for the mathematician, but maybe
Yeah.
this is the reason why I'm here on the course, to learn more and then see maybe what is get out of this. Yeah, thank you so much.
Absolutely. Yeah,
Looking
no, I
forward to
think
the next sessions.
yeah, I
Thank you.
think tomorrow it should be really helpful for that
Thank
type of work. Yeah, absolutely. Thank you, Anna.
Thank you.
Maybe I have one more question, but that's more of like just out of curiosity. I'm not sure if it biologically is really relevant in that sense, but like when you look at the base editing and you look at the single variants, right, in each, let's say, event on its own, have you also, I guess it's also like a huge like it gets kind of exponentially of the combinations, but looking at the combinations of two variants, what the outcome would be. But I don't know if, for example, in certain cases it would be relevant, and you would observe that once you have kind of a certain mutation, let's say in that receptor, that you would have a second mutation if that first mutation is already the driver. But have you ever looked at this, and did you see some, let's say, greater effect when you saw like a combination of those two events? Yeah.
Yeah, that's it's an aspiration really to do so many combinations. I think where the combination field is at the moment, people are still looking at dual gene perturbations, and that's already quite challenging. So, you know, whole genome by whole genome is for just genes is you know, it's seemingly out of reach at the moment. In terms of the number of single nucleotide variants you could do in combination, we haven't really looked at that. Normally, as you say, the oncogenes have a driver mutation, and then we install again another mutation on top of that. That's why we saw some of those drug addiction mutations, but also the drug resistance ones. We would like to look at combinations of variants using functional genomics, but again, yeah, the scale is prohibitive at the moment, and we haven't done that. But there are some papers from the deep mutational scanning fields. They've actually been able to do that at a large scale. So people at the Sanger, like Ben Lehner, he's been looking at, I think there's a preprint from his lab where he's looked at all the combinations of variants in this very small protein-protein interaction between two proteins. And then you can build a map of epistasis, essentially. So when you make one mutation and another, do they cancel out? Are they synergistic, or are they neutral? So that's a very small region of a protein-protein interaction, but very interesting. Would be powerful.
Okay, thank you.